Good day, and welcome to the Patently Strategic Podcast, where we discuss all things at the intersection of business, technology, and patents. This podcast is a monthly discussion amongst experts in the field of patenting. It is for inventors, founders, and IP professionals alike, established or aspiring. And in today's episode, we're providing a multivitamin to help with fortifying your life science patents against the plagues of Section 101 and 112. Life sciences are currently faced by at least two major plagues in our patent world. The first is that many life science innovations have been deemed ineligible in terms of patentable subject matter. In other words, the courts and the patent office believe that the patent laws are not meant to protect these innovations. The second plague is that the courts believe that many life science patents are not enabled. In other words, they're not described in sufficient detail to enable one of skill in the art to make and use the invention. These subject matter eligibility and enablement plagues manifest in dreaded Section 101 and 112 rejections. The practical implications are staggering. When it comes to eligible subject matter, in the last 5 years, over 80% of abandoned life science applications had a final rejection from the USPTO stating that the innovation did not include patentable subject matter based on subject matter eligibility criteria. And as we'll explore, 101 and 112 rejections can surface in a myriad of ways, but no idea is too big to fail. Case law is now littered with patents whose innovators' ideas proved to be revolutionary, but whose patent claims ended up invalidated because they failed to demonstrate in their applications how their observations could be practically applied, or failed to prove that they deserve the breadth of coverage they sought. In this month's episode, Dr. Ashley Sloat, President and Director of Patent Strategy here at Aurora, leads a discussion along with our all-star patent panel delving deeply into these rejections. And in the interest of avoiding a podcast 101 rejection, provided some very practical application tips that will help to fortify your life science patent applications. Ashley is joined today by our always exceptional group of IP experts, including Kristen Hansen, Patent Strategist at Aurora, Daniel Wright, Patent Strategist, David Jackrell, President of Jackrell Consulting, Shelley Couturier, Patent Strategist and Search Specialist, David Cohen, Principal at Cohen Sciences, Amy Fiene, Patent Attorney at Vancott and Adjunct Professor at BYU, and Steve Stupp, Partner at Stupp Associates, LLC. Now, before taking you into the group conversation, as we often do, I sat down with Ashley to discuss a few concepts that could use some extra color and context for those newer to patenting. All right, thanks for joining me, Ashley. Based on feedback we've received, I know these primers that we front load with have been incredibly helpful on-ramps to the deeper conversations that follow, and I know this one will be no exception. A couple of episodes ago in discussing ownership, we explored sections 102 and 103, which deal with rejections around novelty and obviousness. In this month's episode, the rejection focus, if you will, is around sections 101 and 112. Could you talk briefly about each for those who haven't yet been on the receiving end?
Absolutely. So Section 101 deals with what subject matter is eligible for patenting. And generally speaking, this should be essentially everything under the sun. However, it implicitly has been read into this Section 101 by the courts that things that are natural phenomenon, abstract ideas, or natural laws do not are not protectable under the patent laws. And so unfortunately for life sciences, they have basically held that many life science innovations are natural phenomenon, and so they are not eligible for patenting. And so a lot of, what we talk about in today's podcast is how do you better shore up your patent application to be eligible under Section 101, and what can you do? So it can be, as simple as, I think one of the hallmark cases for Section 101 was around fetal DNA, detecting fetal DNA in the mother's blood. And of course, this is amazing technology because it really reduced the risk of the fetus and the mother over, current methods like amniocentesis and things like that. So, but because of how the claim was drafted, it very much centered on the natural phenomenon and didn't, more directly tie in practical applications or things like that. And so a lot of the patents around this technology were invalidated because they were deemed to not be eligible subject matter. So, but in any case, that's Section 101, and probably many, I think other podcasts that we've done also talk about software being a difficult one under this section as well. But as you know, today we're focusing on life science. The other one is Section 112, which has three components. written description, enablement, and best mode. So the written description requirement means that the specification must describe the claimed invention in a manner that is understandable to a person of ordinary skill in the art. So it shows that the inventor actually had possession of the invention at the time of filing. The enablement requirement, which is different than a written description requirement, is that the specification must teach those skilled in the art how to make and use the full scope of the claimed invention. And this is the key part, without undue experimentation. And as you'll hear in today's discussion, that undue experimentation is there's a lot of discussion in the courts around that and what really does qualify as undue experimentation. Is it still undue if you teach one of skill in the art exactly how to arrive at various chemicals that may be covered under your patent, or do you have to actually teach every single chemical, or can you just teach them a process to get to those chemicals, and what constitutes undue experimentation? And then best mode is exactly what it sounds like. You have to describe the best mode. You can't keep the best mode for yourself and only describe those that were not the best. Now, you don't have to label it as the best mode, but you do need to describe it.
All right, so I think that's actually a pretty good segue to the next question. You and the group talk about genus and species several times during the conversation. Now, this talk is about life science patents, but these references aren't strictly biological. Could you explain what genus and species can mean in the world of patenting and claims in particular?
Yeah, absolutely. It is a little bit of a misnomer, but and as it is in classical phylogeny, genus comes before species, right? And genus is a broader classification than species. And so if you think about it in that lens, a genus claim is a claim that covers a broader range of more narrow species, right? So you could have, a broad chemical class, a broad genus class of chemicals, and then more narrow species that fall under that genus. So, think about it from an umbrella perspective. A lot of inventors, especially in the life science, invent one vertical, right? They invent a new chemical, they invent a new drug treatment, something like that. But there may be other drugs that look like that drug, or other chemicals that look like that chemical or behave like that chemical that they believe that they also deserve protection for, right? That they deserve a broader umbrella that covers all those chemicals and all those drugs that look similar. And so in that case, you would say that the broad claim is the genus claim, and the more narrow claims around each chemical or each drug is a species claim. And so what we'll talk about today in the podcast is essentially the death of the genus claim, meaning that the courts are increasingly saying that you need to show some kind of proof of possession, or like reduction to practice, some data or something for each of the specific species that would fall under your genus claim. And if you can't do that, then you don't deserve the broad genus claim. And so this is kind of one of those unfortunate things that's happening as well, that you could fully lead the horse to water, walk them through the exact process that they would need to do to create new chemicals, create new drugs that look like that, behave like that. But because they're in an unpredictable science, because it's, life sciences can, like I said, be unpredictable, can be difficult, the courts have basically said that that's not enough. That's still undue experimentation, and you actually need to reduce to practice at least, one example of each of those species. And so it's becoming much harder for life science inventors to get broad claims, broad genus claims.
Thanks. All right, 2 other legal terms pop up in this episode. They're more in asides and not quite as essential to understanding the thrust of the conversation, but they're still very important in patent law, so definitely worth taking a second to define. The first is just, what is a Wands factor?
So when looking at Section 112, which remember is your written description, enablement, and best mode requirement, or part of the statute, the Wands factors were developed by the courts, and it is a test for enablement. Does the specification and the figures teach one of skill in the art to make and use the invention? And so they walk through several factors to determine if the patent application is truly enabled, or the claims are truly enabled.
And what is an estoppel in patent law?
So this is a general legal principle, but it basically precludes or prevents a person from asserting something today that is contrary to something that they did or said previously. And so, for example, in patent law, during prosecution, which is the bartering that happens between you and the patent office to get your issued patent, if you say that my claims definitely don't include X, and then later in a litigation proceeding or some kind of post-grant proceeding, you say, oh, my claims totally do cover X, that you are stopped from saying that it definitely includes X because you just said it didn't include X. So it basically prevents you from talking out of both sides of your mouth.
Makes sense. Thanks, Ashley. Now, just one more thing before joining the panel. We have some exciting news to share. Back in March, in our Inventor Stories Volume 1 episode, we introduced you to 3 impressive innovators who are all past recipients of our annual RISE Award. This award provides a free provisional U.S. patent application or $5,000 towards a non-provisional U.S. patent application to a selected applicant. This is in recognition of their innovative startup excellence. We received an overwhelming response of applications for this year's award, and the selection process was incredibly difficult. But I'm happy to announce that our selection committee has chosen Hearsight LLC as the winner of the 2022 RISE Award. Hearsight is tackling the important mission of reimagining assistive hearing technology. When we look for ideal RISE recipients, we look closely at potential for societal good, matched with a team that can execute. And Hearsight is just this kind of impactful innovation, backed by the kind of DNA it takes to see through to market success. Assistive technology is a corner of the market that unfortunately still has way too much white space, and we're happy to help fill it in any way we can. Due to the high volume of quality submissions and our desire to help as many as possible, we decided to once again expand the offering beyond the single first place award. The first runner-up this year is Thermoverse. They're focusing their efforts on much-needed solutions that will greatly improve the heating and cooling economics for low to moderate income housing properties. Their innovations are both timely and necessary. Given skyrocketing energy costs, soaring home prices, and housing shortages across the country, disruption in this space is essential to maintain viability to as many LMI properties as possible. And the second runner-up is Spiritfire Aftermarket with their vehicle lighting solution. We love gritty entrepreneurs who roll up their sleeves and find ways to innovate. Spiritfire's story is a great example of finding a real market need, self-bootstrapping, and doing a practical thing very well. We'd love to say more, but we can't, since public disclosure on an IP podcast is beyond forgivable, but do stay tuned. We'll hopefully be introducing you to these innovators in a future Inventor Stories episode. All right, without further ado, take it away, Ashley.
All right. So kind of what we wanted, and Dan's going to jump in here too towards the end with some different thoughts as well. What we wanted to do is kind of jump through and walk through some of the issues plaguing life science patents. And so I kind of named these the first and the second plague. So the first plague is Section 101 issues and the which is patentable subject matter, as you all know. And then the Section 112 is adequate written description. And so, the Section 101 thing has been around for a long time. So I don't think it's necessarily new to anybody, but what I kind of wanted to do is sift through some of the case law and kind of pull out some of the nuggets. But from a background perspective, the main art unit that deals with a lot of these diagnostics methods of treatment, you can see that the name of the art unit is molecular biology, bioinformatics, nucleic acids, recombinant DNA and RNA, gene regulation is art unit 1631. And over the last 5 years, they've had about a 60% allowance rate, which isn't awful, but, maybe could be a little higher, 10 to 20 points. They've had 718 allowed patent applications, 485 abandoned applications. And of those that were abandoned, 80% of them had a 101 rejection at the final office action. So, not a great outlook from that perspective. And so when you look at it, I found this graph through a great website. Actually, we'll put that in the notes. But this is, if you look at, this is the art unit for the last 5 years, that same 1631 art unit. And you can see that there was, about 50% 101 rejections. Obviously, this actually wasn't that bad. They had the references around Alice or Mayo or Myriad, which are a lot of those key cases. You can see that those were relatively frequently referenced in the office actions, and then a little bit of double patenting. But, the biggest issue was those Section 101 issues. And then actually 112 is right up there as well, probably around, 40%, 35%. So that's really kind of thing plaguing the industry. And I think the biggest issue is, how do you delineate when something is just a natural phenomena versus when you're actually applying it in a reasonable way? And then the other part of that is is your written description enough? And I definitely touch way more on the 101 issue and then just briefly on the 112 issue, but there's a really great paper out by Mark Lemley and a few others just in 2021. And I haven't read the whole thing yet because it's, I don't know, like almost 80 pages, but it's called The Death of the Genus Claim. And it's a really good read from what I've read so far, just around the shift over the last 40 years or, 50 years from, companies being able to claim kind of these broader genus claims when they've discovered, a class of compounds that have certain biological properties, to now the court's kind of restricting them down to potentially just one species. And I'll kind of explain why they think that shift is happening, because I think it's kind of interesting. And I kind of saw it happening a little bit, but I probably couldn't have articulated it and the reasoning behind it like they did. And, so it's really interesting. So anyways, we'll touch on that briefly.
What's interesting when you start to see 112s and 101s uptick, they often coincide with examiner training. And so 3 years or so after 112 training, here we are and everything has a 112. Yeah.
Right. Yep. But
not to be cynical.
Right. For sure. But and I think the other interesting thing is the courts. Like, I feel like the courts are, I don't know. I feel like the courts could use some 112 training, and so the courts could use some patent training too, because I feel like they read a lot of things into the statutes that aren't there, which is then impacting, a lot of precedent and how things are drafted, and it maybe not necessarily supported by the original laws. And so I think that's kind of the other interesting thing that's happening. Everybody could use all a bit more patents, right? So nobody's probably unfamiliar with this, but just really briefly, the subject matter eligibility guidance, they basically ask whether your claims are directed to a statutory category, which is process, machine, manufacture, or composition of matter. And, if it's not in that category, it's not eligible. If they think it is in one of those categories, they ask if it's directed to a judicial exception. And as we all know, it's laws of nature, natural phenomena, and abstract ideas. If it's not directed to a judicial exception, then it's eligible. If it is, they ask whether it's integrated into a practical application. If it is eligible. If it is not, they ask whether it has significantly more than the judicial exception. And if the answer is no, it's not eligible. And if the answer is yes, then it's found eligible. So that's generally kind of what examiners walk through. Of course, in the life science, they're going to rely on more of the life science case law, myriad, and things like that. Whereas in the software, they're going to rely more on Alice and other related cases. So then briefly, the second plague, and I'll touch more on this one here briefly, and then we'll jump back into 101 is, and I do think a lot of this is the death of the genus. I think it can really summed up with that statement from Lemley and others. But, where the shift has been happening is that previously, basically what you have to distinguish is whether you're dealing with known unknowns, because I believe if you're dealing with known unknowns, then that's not undue experimentation, right? If a researcher knows how to navigate all the known unknowns, and they should be able to do that to practice the invention. But what you don't want is a whole bunch of unknown unknowns, because then that is probably undue experimentation. And so what, there has been argument around is whether a person of skilled in the art needs to find a species that works in order to practice the claim, or whether they need to find every species that works to make and use the invention. And I think what a lot of experts would argue right now is that the way the courts are interpreting Section 112 is that a person of skill needs to find every species that works. And so even when the spec is super, super clear around what needs to be done to find a species, they say that, well, it still would be undue because there's too many species to find. And so it's a really weird shift that's happening that, you're basically shifting from, the question of whether making and using the invention requires undue experimentation to whether such experimentation is required to define the full scope of the invention by figuring out all the possible species. And so I think that's where a lot of this 112 issue is coming from. So just, briefly, there's a few cases up on the slide, but, the things that obviously, if the field is super nascent and unpredictable and rife with failure, then of course you probably need to have a really, really significant description to support those claims. So I don't necessarily disagree with that, right? And so that was the Enzo Biochem versus Calgene case in 1999. And that was antisense DNA technology. At that time, it was super new. And so they probably did need to describe more adequately in their description, various ways to practice the technology. This one was surprising, even iterative trial and error led by the spec can still create an enablement issue, because this was one of those where it was for rampamycin chemicals for the treatment of restenosis. And it was, if you multiplied all the different R groups that one could have on rampamycin, it was, a huge amount of potential chemicals that could have been created. But, one of skill in the art could have very easily found one chemical that worked, that was within the scope of the claim. But where the court kind of, I think, erred is, again, they thought that somebody would have to find all of them to make the claims work. And I think that's kind of crazy, because, somebody just needs to be able to find one to practice it, right? Because that shows that they can use the spec to their benefit. And then there was another case. This is Idenix Pharma versus Gilead, and described a screening process allowed for straightforward identification of working embodiments. But still, the court said that it required undue experimentation, because, again, this 2-methyl nucleoside for the treatment of hepatitis C virus, they basically, again, said there's so many 2-methyl nucleosides out there. How is any person supposed to, understand the full scope of the claim? And instead of just saying, are they able to practice it? And it's like, well, for sure, because the screening process was very clearly delineated, and it was very much within the skill of, they actually even had experts basically saying it was within the skill of one in the art. So it's, honestly kind of ridiculous. And so, but this is where I think, Lemley and others are kind of saying this is the death of the genus claim, because the courts want somebody to be able to find all of the possible working examples, or all the possible chemicals that fall into a genus, rather than just, be able to practice one. And I think that's an error.
Yeah, actually, I had a question about that. So given all this case law that you've dug into, something that I had always heard about the genus and having good support for it would be like having enough examples in the specification that you have like a cross section of the species. So maybe you'd say if you have a molecule in your genus with like 4 positions for a side group and then 4 options for that side group, you don't need to show or show data, right, for all 16 combinations. But if you did sort of 1 or 2 side groups of cutting across all the categories in some of those positions, cutting across all of them, do you think it would that, based on your reading and what you were just describing, do you think that would still be enough, or are they really saying, no, you need to like have experimental data on all of the combinations?
Yeah, I mean, I think that's a good rule of thumb that a lot of people, and I think Dan could speak to this even more so probably than I could, but I think that's a lot of what everybody's been doing now, is saying, well, you got to make sure you support. But I think where people are struggling, though, is that you don't necessarily have working examples for all of those chemicals. And so if, potentially if they can show at some later date that different methods that you described don't support that class, that could call into question whether you deserve the full genus, right? Versus just a subset. But Dan, do you have additional thoughts?
Yeah, David, that's your tactics there is generally what I've heard, and more or less kind of has done as well, that if you've got all these variants, it's very easy to just name them all. I mean, at this point, chemical software can just spit out all the chemical names. That makes the lists incredibly long. But what's more valuable is to show the cross section, so that you indeed have each type of group in each type of place, as opposed to enumerating each variant of each type of group. But critically, what I have seen examiners do, especially in foreign jurisdictions, the US is still a little bit more flexible than I have found, in particular East Asia, and increasingly perhaps Europe, that your in addition to just depicting a cross section of the compounds, your data also needs to reflect that cross section.
Mm-hmm.
So even if you've only got, even if you showed methyl and halogens in these positions, if you've only got data for halogens, for a halogen, some more strict jurisdictions may not go for the methyls if you've only got data for halogen.
Okay.
So increasingly, yes, I feel like your cross section, the cross section strategy is still kind of what people go for, but the data of what you show in that cross section is increasingly more valuable than just naming them.
Yeah, because like I said, in some of the case law too, David, is that even with different experts stating that the process was like clearly laid out to be able to implement, to identify all these different ones that they had put into the claims, it wasn't enough, because it still would have been like, in order to quote unquote find all of the species, it would have taken a lot of experimentation to do so, rather than just saying, could somebody find one or make one? Well, sure, they could just make one and probably it would work, or they could make 4 or 5 and one of them would probably work, but that's no longer enough. So that's, I think, encouraging there to be some working examples of a few, or one at least in each cross section or something might be advantageous to kind of find what process differences maybe occur for each one.
Yep. Yeah. Okay. Yeah. Thanks.
Yeah. Interesting. So then here's the WANS factors that a lot of times are kind of used for the section 112. And so I'm not going to go through these in detail, but, many of you, maybe everybody is familiar with these, but it's obviously the quantity of experimentation necessary. This is, kind of deciding what's undue versus not undue experimentation. Amount of direction or guidance presented, like how well does the spec teach? Presence or absence of working examples. I've even heard more and more that prophetic examples are kind of not super helpful anymore. The nature of the invention, what kind of art is it? State of the prior art, what was the level of skill, at that time, how much, would they have to experiment to understand the technology? Like I said, the antisense DNA case, the antisense DNA technology was so new at that time, and they just didn't teach enough for how nascent that field was and how much failure there was in that field. So I can kind of understand that case more in the sense that, it was just so new. So you really needed to bolster more support for all the different other use cases or other DNAs that you could make with that technology. The predictability or unpredictability of the art and the breadth of the claims. And so kind of as you're walking through this with clients, thinking about all these different factors and how you describe the technology. But kind of going back to 101 now. So obviously everybody's familiar with the Mayo Prometheus case. So they did describe in the claims the relationship between metabolite concentration and the likelihood of drug effectiveness, but did not necessarily determine or did not necessarily describe using the improved treatment in the claims. And I think, as you'll see from the few examples I have that follow, that really seems to be the critical step, is some kind of almost like treatment step or some kind of, really clear application of the natural phenomenon. So, so for method of treatment claims, a lot of these had an active treatment step. So the Vanda Pharmaceuticals versus Westward and Endo Pharmaceuticals versus Teva, they both had more of an active treatment step. And then this Athena Diagnostics versus Mayo, they had detecting not natural autoantibodies, but again, they were just correlating with the disease state, and there wasn't any step around, okay, so now that we've correlated these things, now we're going to give more drug to this individual or less drug to this other individual, or we're not going to give any treatment to this one, but more treatment to this one. So in Vanda Pharmaceuticals versus Westward, you can see that this claim was found eligible, and you can see this was a treatment method for schizophrenia. And so they do talk about determining patients with a certain point mutation, but in response to them detecting that point mutation in the individual, they then treat the patient differently, better, whether or not that point mutation exists. So if they're a poor metabolizer, then they, have one treatment paradigm. If they're a different kind of genotype, then they have a different treatment paradigm. And so again, that's, you can see that pretty clear language around actual application of what happens now that you've understood what this natural phenomena means. And then similarly in endopharmaceuticals versus Teva, you can see that, again, this is another method of treatment claim. And you can see here that, again, they're providing a solid oral controlled release dosage form and then orally administering it depending on how the creatinine clearance rate was found. So depending on how that was determined to be in the patient. And so again, they measured or determined this natural phenomenon, but then actually really truly applied it from a treatment setting. And so you can see where this falls short in this Athena versus Mayo collaborative, Mayo. You can even see just in the length of the claim is already kind of a little bit of an indicator, but so they're basically determining this muscle specific tyrosine kinase, and they basically just detected different antibodies to it in bodily fluid as a diagnosis for transmission of a developmental disorder. But they never say that, okay, now we've detected this and then we're going to treat them with a certain drug, or we're going to, there was no just active step. And I think that's where they fell short. And so then you'll kind of see that carry through. But so that was, and then so method of treatment is one aspect, but then natural product claims, obviously more and more natural products are coming out and more companies want to protect their natural products. So I think it was also interesting. So there's a few cases into that. And one, I included this one. It's not exactly natural products, I guess, but it's, it's about detecting bacteria. I think the first case was the Roche versus Cepheid, and that was for detecting mycoplasma. And here they talked about a specific set of DNA primers for detecting Mycobacterium tuberculosis. And again, what the courts had argued is that the development of primers was kind of routine and conventional, and the use of PCR was routine and conventional. And so there was no true application of the natural phenomenon, into a new space, no extension beyond that. So maybe if they had to do PCR in a different way because it is mycobacterium, or whether they had to maybe design more unique maybe a different kind of PCR, different kind of primer sets to, accommodate for the fact that it's mycobacterium, or whether they had to isolate it in a weird way, because there was that other Cells Direct case. I can't remember if that's the correct one or not, but where, they had this different freeze-thaw cycle of the cells and then determined viability. But the whole point around the freeze-thaw was to have better viability for later assays. So if even there was something like that in the claim, I think it probably could have pushed them over the edge towards eligibility. But just the pure primers and then routine PCR, isn't enough per this case. And there was another canine case. There was a case around golden retrievers and identifying genetically golden retrievers based on a SNP that they carry for genotyping golden retrievers. And that one was also found ineligible, because it's highly similar to this one. There was also natural alternatives versus creative compounds. This was found eligible. And again, this had an active treatment step, because what the court had said is that when you give this beta alanine to a human, it actually alters their natural state. And so, this results in better performance for athletes, or and so again, it changes, it's an extension beyond the natural phenomenon, and it adjusts their a person's natural state. It's, no longer their natural chemistry, and it has specific dosing requirements. It obviously puts them well above the typical beta alanine dosage in a person. So that's why this one was found eligible over some of the other cases.
Hey, Ashley, I don't know if you were going to touch on this, but these treatment steps seem to have a pretty big implication on who infringes on the claim.
Mm-hmm. Yeah.
Right?
Yeah, because it's basically, we're going to be way more induced infringement, right? Because it's going to be some product supplier, right? Some drug supplier, some natural product supplier who then, presumably has to put in their label, in their, docs, that this is how it's supposed to be taken. And this is, the effective dose and, things like that, and, or some of these more even diagnostic claims, it's going to be some kind of hospital, right, that is running these tests, right? Because I mean, nobody's at home, seeing what the levels of some SNP are to determine if they have schizophrenia. You know what I mean? And so it is definitely, so I think that's an interesting weighing that from a company perspective, weighing the value in a claim that, I think the method of treatment still has a lot of value, but I think it has more value in a portfolio that has claims directed to the actual composition or directed to the actual kit or the system that's being sold that is then used by some kind of healthcare provider to execute on a method of treatment.
Right. Yeah.
Hey, Ashley, just a follow-up on that. Do you have any more detail as to what the extent of a method of treatment is needed? There was that, I forget the exact citation, but there was a case, 2013 timeframe, where they were just doing a threshold comparison based on a metabolite that was during body's processing of a particular drug, and they were using that to adjust treatment. And that was not found to be patentable. That was deemed to be sort of trivial. And so the question is what is there a threshold or a guidance as to what level of guidance for treatment is needed as a claim element to provide patentability?
Yeah, I mean, so from these cases that I've seen, so it has, so this one, again, I remember hearing what you're talking about is a threshold above and below. Do you remember what the actual treatment step was, Steve?
They gave him a drug and it was metabolized, I think, in the liver, and it was a cancer treatment, and they were basically trying to dial in how much to give the patient, and it was just based on a threshold, and it was deemed ineligible.
Because I think in that case it was more still the natural phenomenon. Like they weren't altering the natural chemistry of the person or anything like that. It was purely saying, they're above or below, and then we're going to, treat you accordingly. But it wasn't because they were not altering anything around the natural phenomenon. The natural phenomenon was just a means to an end, is my understanding. Because if you look at some of these other cases, you have in, the Vonda one, they're actually administering a drug, like an actual drug to the patient that allows them to either better metabolize or less, prevent this QTC prolongation of a patient. And so it actually affects the patient's biology. And then same with endo, they are here determining creatinine clearance rate. But again, then the drug amount changes. But I guess that one would be a little bit more similar. Potentially, I'd have to read the claim, that 2013 claim set, but that one could be kind of similar to endo. So I maybe would want to compare that one to this endo versus Teva case in 2019 to see, because I think this one's probably the most similar I've seen.
So, I've seen, changing something other than what occurs naturally in normal biology is one sort of safe harbor, right?
Right.
For genetic material, right? Like there's that one case where you're creating synthetic biology, no problem. Whether or not that's 102 or 103 patentable, it's 101 patentable. If you're making something new that doesn't exist naturally occurring, you're in good shape. And I've seen those kind of cases get allowed without having a treatment. And it looks like, for what you're showing here it's a much richer set of alternatives. There's ranges and different values. It's not just a simple threshold. And I think the issue becomes, for lack of better words, is something analogous to static business logic. If you're always doing the same thing, for every patient, you're not going to be patent eligible. And the closer you are to that,
right,
the less likely you are to be patent eligible. So if you have a simple threshold and you're just looking at the naturally occurring metabolite as something is metabolized, as best I can understand from the Federal Circuit ruling, is they're in essence saying, that's trivial.
You're going to do it, like you said, how would you how would you even regulate that? Or how would you even define, I mean, everybody would be infringing then, that would be in that camp, but maybe that was part of it too.
Yeah. So thanks. That helps.
No, it's a great question. So I'm going to go look up that case now, because then I'll pass it around to everybody too once I find it, because that's really great point, and I think that's some of the parsing that's kind of going on as well in this world.
To kind of build on that, or just like another little example quickly, is that I had a case recently, not in biotech, but in software, where there was a 101 rejection, and the claim that was rejected had a lot of determining and measuring and calculating, and then taking this information into account to determine different courses of action, and that was rejected. But what the examiner pointed us to, and what was eventually allowed, is that there had to be an active step in there. Mm-hmm. So you're measuring things, you're determining things. What do you actually then do? What do you change? What do you sort of affect in the system based on all of this? And so it was a very small wording change. Instead of sort of determining a method for blah, 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 it was determining that and then doing the blah, blah, blah. Like all we had to do is add a little clause. So that made sense to me. This whole adding a treatment step made sense to me, sort of from that angle as well, just where you need to have, like you said, in a way, affect the system, change the chemistry of the person, or whatever, do something active.
Yeah, there has to be like that active step, right? How are you kind of influencing the system, I guess?
To turn this back to machines, it's in software, you have to have an output of some kind. You have to do something.
Mm-hmm.
So it's kind of a weird parallel, but
it is. But it's true. I mean, it's kind of like it's that's, I guess that's the practical application that they're trying to get everybody to do. Yes, I mean, this is obviously some random kind of tricks, but they don't like the extra solution activity, adding the fact that you did it by PCR, or that you're using some kind of like routine testing. That's not going to help at all, because they still, again, believe you're still kind of capturing the natural phenomenon. Again, this kind of gets into the 102, 103 world, because, well, PCR was known, or whatever method you're using was known, and that's more of a 102, 103 issue. But nonetheless, it kind of finds itself in the 101 camp. And then, you don't need to have the insignificant limitations, because they're not going to provide any benefit. And then like general treatments and things like that aren't probably going to help much either. But it has to be some kind of, true effect on the system. This kind of goes towards the computer, side of the world as well. But, how are you actually applying that phenomenon in an unconventional way? So I think you can even think of the, detecting the fetal DNA in the mother's blood. Again, what they were doing was just detecting it, but then maybe what would have helped them is, okay, now that you've isolated fetal DNA, what are you going to do about it? Right. And I think had they maybe couched it more in the way of technical challenges and the avoiding amniocentesis and things like that, maybe it would have had a better chance. And then overcoming, a particular technical challenge, like I said, and then, they're all, I think there's a good recommendation too, to also include lots of different claim types in this area. So methods of treatment, methods of diagnosis, methods of detection, and going over those that are easier first, which would probably be method of treatment, assuming you have some kind of active step, and then maybe going towards the other ones later, especially through divisional practice and things like that, as hopefully the case law changes. But honestly, when I looked through case law for 101 in the life science versus the software, there is way fewer life science section 101 cases. And I don't know if that's like a general reflection of, obviously software is rapidly changing, lots of people have their hands in software, versus the life science. It's a slower moving field. There's, arguably, fewer people that are going to touch life science in their lifetime versus somebody who's going to touch software maybe, or whether that's some other meaningful metric of what that case law has done to those fields, or whether, the cost of kind of pursuing these longer or over a greater period of time in the court system. I don't know, but it was just interesting. Like I said, it had to have been, I feel like, for every one life science case, there was probably at least 3 software cases, it seemed. So it's interesting. So in that sense, it was easier, I feel like, to make sense of it to some degree, because it was less all over the board, but also less examples to draw from, I guess. But anyways, Dan, sorry. Dan, you have something else. Dan, almost
yeah,
Dan.
no, that's all right. So I decided to take a little different spin here on 101 because the eligible subject matter is certainly the dominant topic from that section. It's a fairly recent development, comparatively speaking. For the most part, Section 101 historically has been about whether or not an invention is useful, which is also historically quite a low bar, and so it is reasonable that it gets overlooked. But towards life sciences, the requirement is still there. And as I'll get to the end here, there are a couple places where people can mess up, and it's a bit of an embarrassing mistake to make. And the law around it, I still think merits some discussion. So that fundamental bit about invention must be useful, case law ends up generating 3 main aspects of utility. An invention's utility must be specific, it must be substantial, and it must be credible. So specific must mean it's directed, it's well defined. It is directed towards a particular activity, a particular role. And this is primarily used to deny or avoid general statements. So one of my favorite lines out of the MPEP is in the section where it's like, you can't state that the invention is simply for mere landfill. Can't just take up space. Now, taking up space, though, can be a very valuable thing. So think of all the washers on mechanical inventions that keep things where they're supposed to be, or ballast on ships. That's just weight that takes up space, that keeps balance there. And so indeed, taking up space can be a legitimate use. But the examples I gave there are specific. They're specifically valuable. So you can't just say, oh, it just takes up space. And how this applies to life sciences is you can't just say, oh, these compounds have biological activity and leave it at that. You got to be specific. And you can't just say, oh, you could use this to treat diseases. And I was like, all right, well, which diseases? What diseases? So it must be specific. Your utility must be directed towards something. It must be substantial. So in this is probably the more flexible of these terms that I found while looking into this. And its primary concern is that your invention, its use has an immediately recognizable effect. This most commonly pops up to defeat the sort of hunting license applications of, oh, here's a bunch of compounds that could be useful. We can do some broad speculation that, oh, perhaps this will be useful towards some disease, but we have no actual data. We have no actual support. We don't know. And then this is where, again, it bumps into 112. But the substantial means that from what you've got ready at hand here, there is an identifiable real world benefit that it treats this disease, or even it inhibits this protein. And then finally, it must be credible. This is the perhaps least interesting bit out of the 3, because does your invention, in light of all the evidence you've given, behave according to known and appreciated scientific principles? So credibility only really pops up when people are trying to put in time machines, perpetual motion machines. In life sciences, I saw some examples of using magnetic fields to change things' flavor. You might end up with some, wild homeopathy related type stuff as well. Might show up under credibility, but the MPEP strongly suggests that the credibility bar is fairly low, as long as there's even an inkling, some sort of suggestion. It's like, okay, we'll go along with this. And then as a curious logical necessity, if you somehow fail the necessity or the utility requirement of 101, you therefore fail enablement. If enablement is that you adequately instruct one of skill in the art to make and use the invention, and there's no use for the invention, you therefore can't teach people how to use it. So you will find hand in hand a utility 101 rejection with an enablement 112 rejection. And important for these considerations, particularly towards substantial, is that the USPTO is not the FDA. And this is something that's popped up in a number of cases, although it's at this point fairly old news, that in order for a pharmaceutical to qualify as a substantial utility, no, you don't need to go through the entire process, the entire clinical process of showing that the drug is a commercially viable, safe, effective drug. The mere identification and notably demonstration, at least a token demonstration of biological or pharmacological activity, towards a specific protein that's associated or specific target associated with certain diseases is sufficient. And this has come up again and again and again since the 80s. Some of the earlier language said things as mere in vitro. The fact that you identify activity in vitro has utility towards pharma companies. Whereas I'll say in that last case listed there, In re Brana from 95, there's maybe a bit more telling statement in there about how, due to the expense and duration of full clinical trials, that if drug applications failed utility without or before the clinical stage, that would be a huge economic barrier and discourage barrier to pharmaceutical development economically. So for a practical economic reason, mere in vitro activity is sufficient. And so in each of these cases, the examples, what was generally challenged for most of these cases was the specification stated, hey, here is a target, here's a protein. It's known that this type of molecule can inhibit this molecule, or it can inhibit the target. Here are some new variants of it. Here is literally some data showing in vitro activity. Did they test the whole entire scope? Not necessarily. Did they fully develop the compounds into proper pharmaceuticals? No, but they at least had the background of the knowledge of this specific disease is related to these specific proteins. And here is token evidence. Here is literally some evidence that our class, our molecules can interact with these proteins. And that is sufficient for the utility. So now we'll look at some examples.
Would actually be sufficient for 112, especially now though, because a lot of those early EPO patents, that try to protect variants or different ways of creating EPO had section 112 issues because they didn't enable somebody to practice the full scope of the claim. They may be taught one or two ways to do it, like using some of those assays, but they didn't teach the full scope of it. So I wonder in the current interpretation of 112 whether they maybe would have survived 101 but wouldn't have survived 112.
Yeah, quite possibly. Yeah. Absolutely a legitimate observation there that perhaps indeed it wouldn't be enough to enable the full scope, but for the purposes of determining that specific and substantial utility, that literally something, token evidence is enough as long as it is directed towards treatment of specific diseases, inhibition of known specific biological targets.
So how would you fix that? Would you just have to go extremely narrow if you didn't have enough to support full enablement of everything?
So you're talking about an example where you've got a class of molecules and then you get rejected for lack of enablement for the whole scope.
Right.
Yeah. So that's a hard one to fix. Sometimes examiners are content to listen to, if there's information. Sometimes you can show them data related that ends up in the case file. Not everyone likes that. Sometimes you can find publications that show, the relevant state of the knowledge at the time of the filing. That you got to be careful with that, so you don't end up making it obvious. But sometimes you can play games with that.
Is that like submitted like a 1.132 deck, like an affidavit of inventors, or?
Yeah,
like
you can do you can do affidavits from inventors. That's possible. Being careful with all other publications, that's another process. But it's hard sometimes. And what I have found is, depending on the practicalities of the case, of what's important, and considering your jurisdiction, sometimes it's not worth it to fight it. Sometimes the estoppel that you would introduce in some other, by putting it forward in less important jurisdictions, it's just not worth it. And hopefully you have enabled what is the critical, the golden goose or the golden egg of your genus. Hopefully that's enabled, and so you find comfort in that. But it's a tough spot to be in when you get hit with those enablement bits, because you're generally not allowed to just dump new stuff on into the record. So yep. So here are some examples of where utility failed in life science cases. So this first one, which ended up being sort of the fundamental precedent for pretty much all the cases on the previous slide. Brenner v. Manson from 1966. The patent claimed a novel method for manufacturing a certain subgenus of a type of steroid. Now, at the time, there was no known utility for the class of compounds. The only blurb in the whole application that's actually about what this thing is for is a paragraph that says, hey, these compounds are like these other compounds, and these other compounds are being considered for diseases in which these certain properties could be useful. That was it. So it did not specify a specific type of disease, nor did it show any data that it could inhibit or could interact with the pathways that it mentions. So the next slide, please. The decision here.
What was their motivation to filing the patent then? I mean, did they just put patent behind their name, or?
So this is where we fundamentally, this is the case where like the patent is not a hunting license line comes out of from the decision. And what I think what they were after was that they were related to the lab that was testing this broader class of molecules. So they were like, hey, we found a cool way of making these class of molecules that looks like this other class of molecules, which we're currently interested in. But the state of their technology, the state of their knowledge, didn't actually suggest at all that those compounds would be useful for anything. And they sort of putting the cart before the horse. They wanted to protect that research that they had done before they actually knew it was going to be valuable. And to some extent, you kind of can't fault them for that. I mean, with R&D and pharma companies, you're testing, especially now with high throughput screening, you're testing all kinds. You can test all kinds of stuff. But the decision came back that no, utility is not satisfied by showing that these classes are currently being screened. That alone is not sufficient. And furthermore, a bit more fundamental, that a process is not useful simply by the fact that it produces its intended result.
That's interesting.
That what makes that process, the process, I guess, is valued holistically by the utility in some sense of the result. And so, yes, indeed, simply that because their method indeed produced the compound, but it did not, the compounds were not substantial. So it was, in this case, it was specific. They said, hey, this method makes these specific compounds. So, hey, it's specific, but because it did not promise, it did not show or suggest a real world value, it was not substantial. So then more recently, let's jump forward 40 years. 2005, In re Fisher. This was a case that attempted to claim 5 ESTs, expressed sequence tags, from corn. And ESTs, they're cDNA sequences, which are, I think, notably, it's one of the things thrown about as, oh, well, those are patentable because you take out the bits that don't matter and they don't exist. They don't naturally exist. So cDNAs falls under the patentable subject matter. And ESTs can be useful for a variety of things within genomics. You can help map genomes. You can find which genes are where and what they coordinate to. But in this case, the function and structure of the claimed sequences and their corresponding proteins were unknown at the time of filing. This case just includes a lot of genomics data. It says that they're ESTs extracted from the specific breed of maize. But what these 5 tags did, unknown. Or specifically, well, they name the common uses for ESTs. There's like 7. They list 7, 8. Pretty standard. Dare I say generic uses for what ESTs can do. Oh, of course, you can use ESTs to map where genes are in the protein or in the genome. You can use them to find similar genes in other organisms. But it was completely unknown at the time of filing what they did, what they encoded, and that's really what the Federal Circuit locked in on. Next slide, please. That these uses for these specific ESTs remained hypothetical in general. So although, sure, you could potentially use it to detect variants, mutations. Are mutations in those genes useful, valuable, noteworthy? Completely unknown. And the specification contained no guidance as to why those claimed ESTs were more useful than the tens of thousands that are present in the genome. And then furthermore, there was no data, there was no evidence that those ESTs would specifically, would succeed at doing any of the things like, hey, let's use this to find mutations. There was no such data. So again, similar to that case from the 60s, the court relied on that. This is a sort of another use testing sort of situation. You have a research group that's trying to protect their research, although there's no value yet out of the research. They're still early on, or at least they did not provide what the utility of these ESTs were. And so the next step for them would be, okay, let's figure out what these ESTs actually do. And that is insufficient standing for an eligible patent. You got to tell us what these things do. They must be specific and substantial in their utility to meet that fundamental bar. So yeah, the lesson here is make sure you do have the goal in mind, or at least you provide the context, the substantial context towards what these things are for. And so if you're too early on in development and you're just seeing what you can make in the realm of biotech and the realm of chemistry, just because you can make it doesn't mean it's necessarily useful. And then you fail the forgotten portion of section 101, the part no one talks about.
Well, honestly, even if you find what it does, even if they had found, like in current case, like even if they had found what these ESTs do, it still wouldn't have been protectable from the other side of 101 because there wouldn't have been it would have been one thing if they were like, you genotype for this thing, and then you give more Roundup to the corn versus less Roundup to the corn. or something. And then maybe that active step would have saved him from the other part of 101. So yeah, it's kind of fun to look at these historically, how they've kind of come over time. And yeah, that's interesting. You do forget about the other side of 101. So that was interesting. That was a good historical lesson.
I always like, because even some questions of law that feel obvious or standard now, they maybe weren't always like that. You go poke at the case law, you go do a bit of digging, and you can find a legitimate question in the record. And because, for example, that case from the 60s, there is a dissenting opinion that I didn't read the dissenting opinion, but I'm going to assume it to some extent argues that no, the fact that they could make it, the fact that there was perhaps the amount of speculation, maybe there's an argument in there that the level of speculation was sufficient. But instead, no, that case from the 60s ends up being the grounding precedent for 40 plus years of biological and chemical utility. So even though now we don't think about the utility requirement so much, there was at a time a legitimate question, a legitimate inquiry. And I think it's worthwhile going back and looking at, so you have the fundamental principle at hand and not just the working convenient result.
Yeah, absolutely. Cool. Does anybody have any questions, comments?
I wanted to say something. So back to the genus versus species issue. From the client perspective, that is the financial perspective, it's, when you're facing a question like that, it seems like it might be better just to accept the species.
Right.
Get the issuance, file a continuation, or kick it down the road.
Yep.
It's a tendency, I think, get all up in arms, blah 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 blah. But from the client perspective, please give us a patent. And then the second thing I was going to say is that, if you're wading into an area like this, it might make sense to describe the genus really extensively. And, if you're talking about a structure, maybe you can attach mechanism to the structure. Why does the structure work? And, you're not necessarily going to get the claims that you want, but you're creating prior art that could block others, which is also, important. So those are my ideas there.
Yeah, I think too, what companies have to weigh is how much maybe it's worthwhile spending a little bit of R&D time, at least proof of concepting a few different members of a genus. You know what I mean? Right. Even if it's like just some like really rough in vitro data, right? That we can make this compound, and we said that it, increases insulin production. And look, when we put it into HeLa cells that are meant to produce insulin, we can get a modest increase in insulin. You know what I mean? And just doing a little bit of those studies on a few random different ones to hopefully show that, more of the full scope of the claim, and things like that. But I agree that, getting the species is better than nothing. And so maybe getting the species and then being willing to get a whole bunch of species patents, right? A whole bunch of other ones that are, if you think about yours particular species being the bullseye, and then some of the closest next ones, hopefully trying to get patents on some of those ones. So do you block a competitor in the most effective way with the genus claim? No, but do you still block them through multiple patents, through like all the different ways that they could conceivably get around it? Yes, and maybe a little bit more painfully longer, but yeah.
Right. Anyway, it's a good session. Thank you, Ashley. Thank you, Dan.
Yeah. So no, it's good. I'm glad like I said, this is something I've been meaning to do for a while. All righty. Well, thanks everybody. Have a good rest of your week.
Thank you.
Thanks, everybody.
Thank you.
Bye.
Bye.
All right, that's all for today, folks. Thanks for listening, and remember to check us out at aurorapatents.com for more great podcasts, blogs, and videos covering all things patent strategy. And if you're an agent or attorney and would like to be part of the discussion, or an inventor with a topic you'd like to hear discussed, email us at podcast@aurorapatents.com. Do remember that this podcast does not constitute legal advice, and until next time, keep calm and patent on.